So we're doing stages of pregnancy and a little bit of stuff with DNA and chromosomes. So first off, let's start off with stages of pregnancy. So first thing that's going to happen is when the egg and the sperm meet, the egg is fertilized, and that is going to be called a zygote. Okay, so that is going to be the completion of meiosis 2. Nuclear material from dad and mom come together, and like we said, a fetus can grow from this point on. So the embryo is going to be from fertilization through 8 weeks, and after 8 weeks it will be considered a fetus. So zygote to blastocyst formation. Let's look at the next slide here. So right when the so fertilization occurs, sperms come over and they fight for the love of the egg, and one of the sperms get into the egg, and now it is fertilized. That is called a zygote. Okay, within 36 hours it is called a blastomere. 72 hours we call it a morula, and after 3 to 4 days it is a blastocyst. It is inside of the uterus. So let's check this out. So at about 2 days we're at our four cell stage. Okay, like we said, blastomere. 72 hours we're at a morula, and about 4 or 5 days we are at a blastocyst. Okay, so we get this big cavity formed here with a inner cell mass, and then what will happen is it'll work its way into the uterus and it will implant. It will like burrow a little hole there, and then we're gonna start getting formation. So that's going to be our implantation. Like we said, 6 to 7 days. It takes about 5 days. Oftentimes the reason why women will get when women get pregnant and they don't think they're pregnant because they say they got their period, when they get their period, what happens is they think implantation is occurring. So when that little morula or blastocyst when the blastocyst is implanting, it causes a little bit of bleeding, and some women just think, oh, I had a very light period, is what's going on there. Alright, so we form this thing called the cytotrophoblast and the syncytotrophoblast. We'll look at that in a little bit. And HCG is released, human chorionic gonadotropin. That is very, very important. The reason why is that is what keeps the corpus luteum from degenerating. And if you remember, the corpus luteum keeps up the levels of progesterone, and the progesterone is what prevents you from releasing LH and FSH. Next thing, we'll have placentation, so it's the formation of the placenta. And then we're going to get the gastrula to fetus. We'll talk about that in a sec. Okay, so here's our zygote. A couple days, we're at our 2-cell stage. We go to the morula at 3 days. 4 days we're at the blastocyst and we're working the way into the uterus, and at about 6 to 7 days implantation occurs. Implantation can take 5 or more days, and during implantation you can have a little bit of bleeding, and some women interpret that as they're having a period, so they don't think they're pregnant. So here's what's happening with implantation. That blastocyst implants and it's growing here. Okay, when that is happening, we're going to have our cytotrophoblast, which is here. Okay, and we're going to have our syncytotrophoblast, which is here. And then we're going to be getting maternal blood vessels. So we're going to be getting blood supply coming to this place. Next, we're going to have these guys, epiblast and hypoblast. Epiblast and hypoblast. What those turn into is right here. Those turn into our dermal layers. We get an ectoderm, a mesoderm, and an endoderm. We will talk about these dermal or germ layers in a minute. These are going to be the layers that are the ultimate stem cells that build everything else in your body. This is about 4.5 weeks of an embryo, and what you see here is we have the yolk sac, we have the amniotic cavity forming, and we have our chorion and chorionic villi filling. Okay, so we'll have our umbilical arteries, and what those guys will be there for is those will be bringing blood to baby, fresh, oxygenated, nutrient-rich blood from mom to baby. Okay, so we went zygote to blastocyst. Now we're going to go through gastrulation, or gastrula to fetus. So we said we had the formation of the amnion, the yolk sac, the aletinoids, and the chorion. The yolk sac is going to be helping out with red blood cell production in the fetus. Okay, the amnion is going to be there as a protective cavity to hold the baby in and to protect it and to help keep temperature homeostasis for the baby, for the fetus. And then in our gastrulation, that is the formation of our germ layers. So you remember we said the endoderm, mesoderm, and ectoderm, and I said this is where all of the germ layers come from, or all of your cells come from. This is the ultimate stem cells. And then next we're going to have our organogenesis. It starts at about 3.5 weeks, and that's going to be when blood vessels form and you get the first fluttering of a heart. So here's your ectoderm, mesoderm, and endoderm. There's a list of stuff that each one of these forms. I like to think of it real simple. Ectoderm does your nervous system and your epidermis. Everything that is pain sensitive is ectodermal in nature. Endoderm is your epithelial linings of the GI tract, respiratory, and urogenital systems. And then the mesoderm is going to form all other stuff. So let's go through our fetal development. This is going to be at 8 weeks, you're going to turn into from an embryo into a fetus. So there's going to be rapid growth, and body structures are going to be becoming developed and appearance. So at about 8 weeks, huge head. These are called limb buds. So you have limbs, but they're not very well developed. All right. About 22 millimeters long, or less than an inch long, and 2 grams. About 3 months, the head starts to shrink down a little bit. You're starting to get digits. You're starting to get some facial features, okay? And you're going to be about 90 millimeters. So you'll be close to the 3-inch range, a little over 3 inches. 13 to 14, the brain starts to develop pretty well. Your cerebellum starts to come in place. You start getting general sensory organs. And you're going to be getting your general sensory organs and the eyes and ears and all that, so you can start getting some sort of facial recognition, or facial features and facial expressions going on. They start to look kind of human. At this point, they're like 140 millimeters long. At about 5 months, you get this thing called the vernix caseosa. Anybody who's had a baby, when the baby is born and it looks like it has cream cheese on it, that is what it is, the vernix caseosa. It is fatty secretions of the sebaceous glands, and it's covering the body. Okay, so it looks like cream cheese on the body. And you get the lanugo. Those will be silk-like hairs. Those are the little hairs those are the baby peach fuzz that babies are born with. Okay, those don't last very long. They fall out pretty short term after birth. They begin to get into fetal position because they're growing and they're getting large compared to the area that they're in, so they start to curl up in their fetal position. All right. About 6 months or so, they're going to grow a lot. Baby grows a lot, mom grows a lot, right? After about 28 weeks, the baby is considered to be able to live premature. It's not always 100%, but it's much more likely. Big issue with it is temperature regulation, and lungs aren't ready yet. So the hypothalamus and the type 2 alveolar cells, the surfactant producing cells, aren't ready yet. So baby needs to be kept under a heat lamp to keep it warm, and also they're going to be needing help with oxygen delivery. Myelination of the cord begins, and the eyes are open. Distal limb bones, okay. Skin is wrinkled and red. Fingernails and toenails are present. Body is well leaned in proportion. All right, so the baby's getting close. It can survive at this point. At about 30 to 40 weeks, the skin's going to be whitish to pink. So if you have ever seen a premature baby, where were we at here? It says the skin's like really pink. Skin is wrinkled and red. So when you see a really preemie baby, one of the features that you'll notice is they're really, really wrinkly and their skin's red. As the baby gets a little bit older and a little bit fatter, the skin turns more whitish or pink. Okay, and they grow quite a bit too at that point. So the effects on mom. Any pregnant woman should know this stuff. So reproductive organs, increased vascularization and engorge with blood, breasts enlarge, organs press against the diaphragm, and you get this release of stuff called relaxin. That is going to make your joints more lax, and it's going to help with the opening of your hips. You get the release of human placental lactogen and human chorionic somatotropin. What these guys are going to help with is production of milk. Alright, and then physical changes. When you think of a pregnant woman, this is what you think of. Morning sickness, heartburn, constipation, frequent micturition or urination, increased respiratory rate. Why? Because all those abdominal organs are pushing up on the diaphragm and it can't flatten out to really get oxygen to get air into the lungs. And you're going to have increases in blood pressure and blood volume. So before baby is born, at about 4 months, that uterus is growing. Alright, 7 months, 9 months. Okay, with patrician, that is going to be initiation of labor. So what happens? You get spikes in your estrogen levels. When you get spikes in estrogen levels, those cause your myometrial cells. Those are going to cause the cells of the uterus to form oxytocin receptors. When those guys have oxytocin receptors, what happens is the whole time fetal cells can produce oxytocin. Alright? So if these uterine cells have oxytocin receptors and fetal cells are producing oxytocin, what that causes is contraction of the uterus. Alright? So both uterine muscles stimulants. Alright? Posterior pituitary gland will begin to release oxytocin, and it works in a positive feedback mechanism. So fetal cells are releasing oxytocin, posterior pituitary is releasing oxytocin. We have high levels of estrogen, and that's making the uterus contract because it's going to be form its oxytocin receptors. So all of a sudden we get waves, rhythmic waves of uterine contractions. So that's your contractions. Alright, and what's happening is the waves are because the waves of the release of oxytocin from the posterior pituitary. Okay, so stages, dilation. This can last for a long time. This can last for weeks. So when you start getting Braxton Hicks contractions, that is the uterus just contracting down and starting to flatten out or efface the cervix. Alright, it is from the onset until you are fully dilated in your water bursts or your amnion ruptures. Expulsion, full dilation till delivery. This usually isn't very, very long time. And then placentation. It's about 30 minutes after baby has been born, and you're going to be detaching the placenta, and this will be your afterbirth. Okay, so with inheritance, some terms you want to know: homologous chromosomes, alleles, homozygous versus heterozygous, dominant versus recessive, genotype, phenotype. So homologous chromosomes are going to be 2 chromosomes that make up a pair in the diploid cell from one from the egg, one from the sperm. So we have 46 chromosomes. One set of chromosomes is from mom, one set of chromosomes is from dad. So each one has a number, 1 through 23. Okay, in pair number 1, you get one for mom, one for dad. That pair number 1 are homologous chromosomes. They code for the same stuff, but they're carrying half your information from mom, half your information from dad. Alleles are going to be 2 matched genes on the same locus on homologous chromosomes. All right. So if you guys have heard of Punnett squares, okay, the way these things can work is they can be dominant or recessive, and they could go homozygous or heterozygous. We're going to do an assignment with these, and we'll chat about this in lab. This is easier for me to draw for you on the board. Okay, and then genotype. So you have your genes and you have your physical characteristics. Your genes are what are going to be the genes you're born with. Your phenotype is going to be your physical characteristics that you display. All right, so here's our karyotype. The 1 and 2, these would be homologous chromosomes. Sorry, 1, there's 2 chromosomes here. These would be the homologous. This pair would be homologous. So each pairing of chromosomes are homologous chromosomes. Okay. During meiosis 1, what's going to happen? We're going to show it on the next page. Tetrads line up, and we get this thing called crossing over. So our chromosomes line up, and what happens is chunks from one chromosome mix up with chunks from the other chromosomes, so they can cross over and they can switch out genetic information. So that's why you're not looking exactly half like dad and half like mom. You look a little bit like dad and a little bit like mom, and you didn't get an exact copy of one of dad's chromosomes or an exact copy of one of mom's chromosomes. You got a mixture of both of theirs. And then with our types of inheritance, we're going to have different types. You can have dominant, recessive. So with dominant, recessive, if you're looking at like with dominant recessive, if you're looking at something, you can be, let me look at some examples of dominant recessive here so I could give you a good one. Let's use double jointed thumb or tight thumb ligaments. Dominant gene's going to be double jointed, tight thumb is not going to be double jointed. So either you're double jointed or you're not. There's no ifs, ands, or buts. With incomplete dominance, that would mean that you're going to have two things kind of sort of sharing. So let's say we took a red cat and we took a white cat, and red cat and white cat have babies. They're going to make pink cats. So that would be kind of incomplete dominance. With dominant recessive, red cat, white cat, let's say that red is dominant over white cat. Red cat and white cat have babies. All the babies are going to be red cats because the red will dominate over the white. But all of those babies will be carriers. We'll talk about that in lab. Multiple allele inheritance. So what that can mean is that you have multiple dominants. So if we have a red cat and white cat and they have babies, they'll be red and white spots. So both will be expressed. Sex-linked genes. These will be genes that are only carried on the sex chromosomes. So females are XX, males are XY. So these will be carried only on the sex genes. 1 through 22 are your normal chromosomes. Your sex chromosomes are set 23. These normal chromosomes, 1 through 22, are your autosomes. So autosomes are sets 1 through 22. Set 23 are your sex chromosomes. And then polygenic inheritance. This is most stuff you think of. This is stuff where there's a wide variety of change. So hair color. You can have light brown, dark brown, and everything in between. You can have light blonde, dark blonde, everything in between. You can have red hair. You can have all different kinds of hair color or skin color or eye color or height or weight or build of muscle. So this is going to be where there is a lot of difference from person to person. This is what gives us our huge variation, are these polygenic inheritance traits. Okay, here's some examples of dominant versus recessive traits, and like I said, we'll talk about these in lab. We'll do some Punnett squares. Okay, other factors. So these can include environmental factors, regulation of gene expression, and mitochondrial inheritance. The big one is environment. It's nature versus nurture. So are you born this way, or has your environment gotten you to be this way? And with a lot of some things are 100% gene expression. Other things are going to be, some things are you're just born that way, and other things are going to be 100% environmental, and then there's still going to be other things that are a combination of the two. Okay, maternal inheritance, like I said, we're going to talk about Punnett squares in lab. That'll help with the other stuff.